So convergent and divergent evolution, they are very different from each other in terms of how they occur, right? First of all, convergent evolution is basically talking about two species that are very far, basically distantly related with each other that somehow evolve the same trait. So thinking about bats and birds, they are very far in terms of their evolutionary trees in their phylogenetic trees, but they both managed to evolve the trait of flight. So that is the meaning of convergent evolution, where there are similar selection pressures that have selected for that same trait, and as such, they both evolve that trait. We looked at this very briefly in the other lesson, and I said that it's not that important, but it's actually still important because they can still ask you questions in your BCE regarding that. So make sure you understand the difference between convergent evolution and divergent evolution. So an evidence for convergent evolution is the analogous structures that we talked about in the last lesson, right? Where they have structures that serve the same function, right? Basically, the trait is to provide flight, and all of these different animals have that same trait, but they all evolved it due to similar selection pressures, but they are all not related to each other. In terms of their evolutionary relatedness, they are very far from each other, and yet they evolved that trait due to similar selection pressures. That is the meaning of analogous structures, and that is an evidence, a physical evidence of convergent evolution. Basically, they are converged towards the same trait, right? Today's lesson is going to focus heavily on divergent evolution. So this is a process in which ancestors evolve into two or more different species, right? So basically a similar ancestor, they started to accumulate genetic differences, and then eventually they speciate into two different species. That is an example of divergent evolution. And what is the physical evidence of that occurring is homologous structures. This is going to be very important for today because that's how we're going to be heavily relating it back to the phylogenetic tree. Nick, does everyone have one already? Okay, so this is basically in line with the natural process of speciation that we've discussed, and also how it works is that they basically accrue genetic differences, and eventually they accrue enough genetic differences to be considered as different species. For example, like we talked about here, right? In the previous example, in the previous lesson, we talked about there being a common ancestor between chimpanzees, bonobos, and the human genus, and then eventually they accrued enough genetic differences to diverge from each other to become different genuses. As such, this is an example of divergent evolution. They are coming from the same common ancestor, and then they are separating out, diverging from each other to become their own species or their own genuses. Okay, this one is not really what's the word? Accurately showing the human genus. It's just summarizing it as humans, bonobos, and chimpanzees, but it's a very simplified example of divergent evolution. So make sure you guys are able to tell the difference between one and the other. All right. So far we've learned about all these different things. We're going to go very deep into divergent evolution today, and also how they relate to phylogenetic trees. So we're going to be working on drawing a concept map of all this, but we're going to be doing this in tomorrow's lesson, not today. So homework for tomorrow, I guess. So today's lesson, we're going to focus on looking at phylogenetic trees. These are trees that are basically illustrating everything we've learned in the previous lesson, right? So in the previous lesson, we are using different evidences to study relatedness of different species, right? We looked at the structural morphology and also the molecular homology. So these are the two ways that we can study the relatedness between different species and find how they are related to each other. And now that once we have that data of how they are related to each other, how closely they are related to each other, we can start to create a illustration, a graphic illustration of that in the form of a phylogenetic tree. All right, so let's look at this first question, right? So did the Tasmanian tiger, now extinct, and the American gray wolf evolve independently of each other but show similar physical structures and hunting behaviors? The similarity between the two organisms is most likely a result of what we're looking at. Yeah, what do you think? Okay, so the key word here, the key phrase here is that they evolve independently of each other. So, sorry. C? D. Yeah, so yeah, basically that's the only logical answer because the fact is they are in very different environments, so they have no relatedness for a very long time. They're very far apart and they have no interactions at all, and they've never had any interactions because one's in Tasmania and the other one's in America. So the fact that they evolved similar traits shows that there were similar selection pressures acting on them to evolve the similar traits, an example of convergent evolution. All right. So today we're going to learn how to use and interpret a phylogenetic tree as evidence of relatedness between different species. Okay, your glossary task. And so first of all, let's start off the basic, right? What is a phylogenetic tree? This is basically a diagram that shows the evolutionary relationship between different species. Basically, how are they related? How far? How did they diverge? How did they diverge, and what kind of species are closely related to them? We can derive a lot of information from a phylogenetic tree, and we're going to be looking at that as well. So essentially, by being able to understand the evolutionary relationship between different species, we can better understand how life has evolved over time and how we've lost and gained traits through that process as well. So how do we construct these phylogenetic trees? Basically, we need data to be able to construct these phylogenetic trees, and that's where the previous lesson comes in, right? We basically collect data in terms of their homologous structures, in terms of the vestigial structures, in terms of the molecular homology as well, to be able to better understand how different species are related to each other and at which point they would have possibly diverged from a common ancestor to become two different species. So by understanding how species are related with each other, by doing all that research in the previous lesson, we can then have a lot of data to be able to construct something known as a phylogenetic tree to represent all the information that we found. So it can be very useful in identifying the evolutionary relationships between species in a very easy way so that everyone can have a look at it and basically derive information about which species are related to each other, at which point did they diverge, and also how related they are and what are the shared characteristics of the different taxa. Taxa are basically these different species, right? Basically, taxa is another way of referring to these different species. And we have another name for them when it comes to phylogenetic trees. We'll be looking at that as well. So if I ask you, when did sharks diverge from ray fin fish? Sorry. By looking at this phylogenetic tree, I ask you the question, right? When did sharks diverge from ray fin fish? What do you think? Can I get Emelinda? Where do you think sharks would have diverged from ray fin fish? Follow the phylogenetic tree, trace it backwards, and look when they would have diverged from each other at this point, right? So basically, that's how you should be looking at a phylogenetic tree. You go from the species and you go work backwards until you see where they would have joined together. Okay, so this can get a bit more complicated to use and to do, But eventually, once we get some practice in of how to do it, it will make more and more sense, right? Next question. How are amphibians and primates related? Amphibians and primates. Okay. If you look at it here, amphibians and primates live with each other. Again, go backwards, right? Keep going backwards, trace backwards to see where they would have related with each other. Essentially, all of them have 4 limbs, and then there was a divergence of the common ancestor to have different traits. One formed to be amphibians, and the divergence created other divergences to eventually create your primates. But essentially, all of these have homologous structures which are relating back to the 4 limbs, right? And lastly, what are the shared characteristics between rodents and primates? So essentially, when we're looking at a phylogenetic tree, we can even relate it back to the homologous traits and structures that they have. If I ask you what is a homologous trait between primates and rodents and rabbits, what do you think it would be? Hair. Basically, fur. They both have fur. That is a homologous structure or a trait that is shared between these two. So this is where homologous structures come into play. You are basically able to find out how related different species are by relating it back to the homologous structures that they share, right? Basically, if I ask you which of these species are vertebrates, all of them will fall under. But if I go a bit deeper, ask you which one has bony skeletons, only these would count. If I ask you which have 4 limbs, only these would count. Amniotic eggs, only these would count. Hair, only these would count, and only these have 2 post-orbital fenestrae. Okay, so homologous traits can allow us to better understand the relatedness of different species, and that will actually come in handy when we are trying to construct our own phylogenetic tree. So we're going to look at that as well. But before we get into that, let's try to understand what are the different characteristics of a phylogenetic tree. Okay, there are 4 main characteristics that you guys need to be able to identify, and we're going to be doing that as well. So first one is your root. Okay, that is basically the common ancestor that is shared between the different species we're going to be talking about. Basically, the line, the origin, what starts everything. Okay, so this point is where the common ancestor started, the first diverge, create this phylogenetic tree. And we will never refer to the original phylogenetic tree because it's too dense. You will always get examples which are basically this size, which always starts with a starting species, or they won't even show you a starting species, they'll just show you a line here. But this is referred to a root. This is basically the last common ancestor shared by all these different species. All right, that if that is the root, the next thing is the node. The node is basically where the common ancestor diverged to become into different lineages, right? So this happens at this very point where the line breaks into 2, right? That is referred to as the node because that shows that represents the divergence of a species from a common ancestor to become different lineages. And then every time it breaks up into 2 different branches, that is referred to as a node. So you can see it here, this is a node, this is a node. There are multiple nodes in any phylogenetic tree. You are just looking for where the divergence happened. That is what you would refer to as a node. When it comes to a branch, it's not really very hard to tell. A branch is basically any line in the phylogenetic tree that is going towards the taxon or the leaf, right? Basically, any of these lines can be referred to as a branch. The most important features of the phylogenetic tree that you definitely need to know are the nodes and the leaf, right? Because that is going to be very important for you to be able to understand how to read one. Okay, the last one is the leaf, right? This is basically the end of any branch. That's where we're trying to get to. This is indicating where the present day or extinct species are found, right? Basically, this could be either labeled as a species name or as a taxon, right? Basically, most of the time you're going to deal with them giving you a species name there, like you have here, right? So if you're having trouble thinking about the nodes, just think of it about remember everything we learned about in terms of speciation, right? There was an original population first, and then they accrued genetic differences, and then suddenly a new species emerged from that original population. So out of one came two, right? That's what's happening at those nodes, right? Basically, one line becomes into two different lines in these nodes. Right here, for example, you can see, follow along with me. So the branch is following along. This means the branch is basically showing how that population is thriving over time, right? Over many thousands of years. And then eventually they've accrued enough genetic differences within that population that they diverge, right? They speciate into two different species. So this is the node specifically because that is where they diverge from each other, from the common ancestor to become into different lineages. So as long as you're able to identify these structures, that's the most important part. So you can actually start to make sense of a phylogenetic tree more if you keep practicing being able to identify different nodes. Okay. So when we think about a phylogenetic tree, especially when it comes to your exams, they can actually give it to you in a few different variations, right? One is the most common variation that we've seen so far, where there's a root starting and then it breaks up and it shows a very structured way of going. Another one is more of a, I don't know how to describe these. Basically, they just look different, but in terms of what their features are, they're exactly the same. It always starts with a root, right? Always go to the very start, and that is your root of any phylogenetic tree. Almost like the tree of a trunk, right? It has to start somewhere, so that's the root. Okay, and then the first node appears, then it starts to further diverge from each other later in the phylogenetic tree. So every time a node appears, there's more divergence happening, and then we will have our taxon or leaves at the very end of the phylogenetic tree. Okay, so that is the end of the branch, indicating where the present day or extinct species are found, and we are labeling that either as species or with the taxon, which are A, B, C, or D and E. Okay, any confusion here, it's going to be very important for you to be able to remember these three features of any phylogenetic tree, right? The root, the node, and the leaves. When it comes to the branches, you can just simplify it as being any line within the phylogenetic tree that is part of the branch already. But the nodes are the most important part, followed by the root and the taxon or leaf. Okay, moving on. So once we start to be able to read or understand the features of a phylogenetic tree, we now need to be able to derive information from it. What can we actually understand from looking at what we're seeing here? So now let's try to break it down, right? So let's look at the human leaf, basically this part, the human leaf, and work backwards to find the first node for the human leaf, right? So if we look backwards at the human leaf, we work backwards in the phylogenetic tree, we can find our first node right there, right? Specifically, let's zoom in. So what you see is that if we work backwards from the human leaf, we go backwards, we see our first node, and that node is shared with monkeys, right? What does that indicate is that we have a we have a common ancestor with monkeys. That means that monkeys and humans are very closely related with each other. That is how you should be reading notes in a sense, is basically to determine how closely related species can be is determined by how far the nodes, the most closest node that they share, right? Humans and monkeys have a very close node with each other. That means they are very closely related with each other. Okay. So that is when we're looking at humans and monkeys. If I ask to compare humans and sheep, we will work backwards for both of them and see where the node that they both share is, right? So we work backwards from sheep and we work backwards from humans, and we realize that the node appears here, right? Basically at node B. That is further than node A. So what we can derive from that is that humans and sheep are closely related to each other, but not as closely related as monkeys and humans, right? So basically it's a comparative thing in terms of how close the most nearest node is between each other. So node A shows a divergence of a common ancestor of monkeys and humans, meaning that monkeys are the closest ancestor to humans because they have the closest node between each other. If we then look at the node before that, we look at node B showing a common ancestor between humans, monkeys, sheep, and dolphins. That means they still do share relatedness, but not as much as with humans and monkeys. So in terms of relatedness, you would go further and further back. The further you have to go back in the phylogenetic tree. Okay. So again, look here. Node B shows divergence from ancestor A. Basically, ancestor A is this one, the common ancestor that diverged to become humans and monkeys. So node B shows the divergence of a common ancestor to become ancestor A, and also another ancestor for dolphins and sheep. So they do share relatedness, but it's just further related, like it's distant relation compared to monkeys and humans. Who's confused here? If you are, please let me know. We can go through it a bit more, a different approach. All right, so let's look at your own phylogenetic trees, right? So when you're looking at your phylogenetic trees, let's look at primates and rodents, for example, right? If you work backwards from primates and rodents, you see that first node. That means that primates and rodents are very closely related to each other because their first node between each other is very close to each other. So that means they are very closely related. But if I ask you to compare crocodiles and primates, their node appears much later down here, which means that they are further related to each other, but they are still related to each other. But if I ask you to compare sharks and primates, that's when you start to realize that if you go backwards and backwards, eventually there will be a point where it is diverging very early on, right? So sharks and it's really backwards until you see a common node between each other, which is right here at the very start of the phylogenetic tree, which means that their common ancestor diverged a long time ago. So in terms of relatedness, it's very little relatedness between each other, between the two different species, because their node happened very early on. Wait, yeah. Okay, so if you need a very easy way to understand this, is basically just say that the further the node that they share, the further their relatedness is going to be, right? So the further back you have to go to find a common node between the two different species you're looking, the further their relatedness is actually going to be. Moving on. Okay, so yeah, like I said, right? You can represent them as taxon. These are your leaves. The branches are where they lead to the leaves. Node is where the lineage diverges, and your root is where the last common ancestor that all the leaves shared are. Basically, the very start of the phylogenetic tree. Also, there's one more feature of phylogenetic tree that you don't necessarily have to identify, but they might be able to ask you is the outgroup. This is basically the species that diverge from the ancestor, the root that has no really, there's very basically that has very little relatedness between all the other different species within the phylogenetic tree. They basically are a completely separate lineage on their own, and they don't share any relatedness between the others because they diverge from the root early on, and they were basically separated that whole time. Yes. Yeah, in this example, that would be the sharks that you're looking at, right? They basically diverged from the common ancestor the last common ancestor very early on, and they never uh, basically never shared any relatedness between any other species on the phylogenetic tree. That makes them the outgroup within that phylogenetic tree. Okay, so moving on. So when we start to put more information into a phylogenetic tree, sometimes they can even give you a time scale under the phylogenetic tree, which also indicates in terms of when these species would have diverged from each other and how long did these species exist before diverging into different species as well. So an example, let's start from here, right? We have our root, which is our last common ancestor shared by all these different species. So hot. Okay. So if we look at that, we can see that the root is almost 12 million years ago. And then the first divergence, the first node appears where it diverged into different lineages at around 11 million years ago. And we can see our first outgroup, right? The black bears, they just did their own thing. They never joined together with the other species. That is your outgroup. And then we see more nodes appearing within the phylogenetic tree where it diverges again at around 10 million years ago and so on. So the further down the phylogenetic tree you go, the more closer you come to present day or closer to, further ahead in time. So the further back you see towards the root, the older it's going to be. So in terms of the root and its divergence, that's referred to as happening very early on. So the root, that divergence happened very early on. And that's why when we try to show relatedness, we say that the closer they are to the first node, the more related they're going to be, and their divergence would have happened much more recently. If we try to relate dog and black bear, we can say that they share very little relatedness between each other because the two different species diverged very early on, about 12 million years ago, to form different lineages, right? So you have to be able to express ideas of relatedness and also express ideas of when they would have diverged in terms of timeline. So you can hypothesize that they diverged from each other very early on because they appear much closer to the root, further away from the closest node. Any questions so far? Okay, very quiet as always. Okay, yeah, this is supposed to be here. Okay, so basically, we've looked at a lot of different things so far in this area study two. We've looked at population genetics, we've looked at speciation, and now we're looking at divergent evolution, and then we're looking at it in terms of a phylogenetic tree. The idea here is to be able to relate all these things together to have a understanding of how this phylogenetic tree is describing that. So let's start off, for example, with tetrapods, right? So a species of tetrapods exists, right? Primitive tetrapods exist. They have a huge population, right? There exists natural variation within that population, and through the mechanisms of evolution, they start to have very, I would say, populations which are being selected for due to different selection pressures, right? eventually those selection pressures cause the different populations to have so many genetic differences that they diverge to become different species. And over time, this process occurs many times to diverge to many different forms of life. So if we try to relate everything we've learned so far, an example of how you could describe it is that a population of primitive tetrapods experienced the mechanisms of evolution and eventually accrued enough genetic differences to cause them to speciate to diverge in the phylogenetic tree, and that gave rise to the ancestor of Western clawed frogs and our ancestors. So basically, when it diverged, we can see that it formed into two different lineages. So that is the lineage for a common ancestor for Western clawed frogs and Chinese brown frogs, and also for us and every other species on this part of the phylogenetic tree. So that is being able to relate everything we learned so far into a huge picture. It's going to be hard unless you can see the big picture of everything we've learned so far in the area study. So you guys definitely need to do your own revision of going back and understanding everything we've learned in this area of study as to be able to draw a bigger picture of everything to relate them together. And that will actually help you better understand the entire topic because they all come into play here, right? The mechanisms of evolution, natural selection, mutation, genetic drift, gene flow, and they work together to cause evolution, and that causes speciation to occur. Eventually, speciation can be tracked over many millions of years to show a huge divergence of evolution between different species, and we track that using the phylogenetic tree, which is showing evidence of that as well. Okay. We'll come back to that in another day, just so that it can be better understood. So now that we've learned how to read a phylogenetic tree and describe the features of one, we also need to be able to make our own phylogenetic tree using the information that they give you sometimes, right? So the key to constructing a phylogenetic tree is basically, like I said, identifying the homologous structures or traits that they share. Basically, structures that are similar but function that can possibly function differently between each other, but their structures themselves look similar, right? So, for example, we're looking at four different classes of animals. One is placental mammals, one is marsupials, one is amphibians, and one is bony fish. We're going to be using these four classes to make a phylogenetic tree. So now what we need to do is look at the homologous structures that they share. So when we look at a bony fish, they have no homologous structures for forelegs and fur. Okay, that is information that is going to be important. For amphibians, they have one homologous structure, but the other one they don't. And for marsupials and placental mammals, they share the same homologous structures. So in terms of that, we can actually start to derive information in the sense that the presence of homologous structures is reference to their relatedness. If they have more homologous structures between them, that means they are going to be closely related to each other because they would have only diverged from each other much more recently because they share more homologous structures. Think of it like this: between Homo sapiens and Homo neanderthals, we basically share a lot of homologous structures because we are very closely related with each other. So we're going to be if we're going to be thinking about where we appear in a phylogenetic tree, we're going to be very closely put together, right? We're basically, in terms of phylogenetic tree, we're going to be almost like the crocodiles and dinosaurs and birds because we would be very closely related on the phylogenetic tree. Okay. After that, we look at amphibians versus marsupials and placental mammals. We understand that the amphibians have only one, while these two have two. That means that they do share relatedness, but just not as much as marsupials and placental mammals. So that means they amphibians diverged a bit earlier compared to them. And we are basically deriving information from these facts, right? First, like I said, amphibians, marsupials, and placental mammals structures or traits, meaning that they are closely related between each other. Okay, we get that information first. They do share homologous structures, so they are closely related to each other. The other information we have is that bony fish don't have any homologous structures or traits with the other species. This means it has the furthest relatedness with the other species in terms of how related they are. It's the furthest because it doesn't have any homologous structures. Another information we know is that that means that it diverged from the other species much earlier because it's basically almost like an outgroup where it diverged so early on that it doesn't share any homologous structures. Okay, and the first one, the fact that those trees share any homologous structures means that they diverged from each other somewhat recently or more recently with each other. Okay, and then we go further into our analysis where we look at amphibians versus marsupials and placental mammals. We notice that they have only one homologous trait or structure in common with each other. That means that they shared a common ancestor between each other that diverged from each other. And most importantly, we see marsupials and placental mammals. They share both the homologous structures, thus making them the most closely related species within the phylogenetic tree. So now we have our 4 pieces of information. We can start to construct our phylogenetic tree, right? So the first thing, right? We know that bony fish has no homologous structures. So we do that part first, right? So we basically lay it out. First, we know that the root is here. The outgroup would be bony fish because they share no homologous structures. So they diverge much early on and they were basically an outgroup. They never interacted with the other species. And then we can put all the other 3 into one, basically because they share even one or more than one homologous structure, so they are all going into another line. Okay, that's our first point. Separate the outgroup from the related species. Okay, moving on from there, we start to put more detail into ours based on which one has the the least homologous structures, which is amphibians versus marsupials and placental mammals. So we know since amphibians only share one homologous structure, that means it would have diverged at some here, right? So that's what we indicate in our next drawing, right? So we show that they were related to each other along this branch, but the fact that amphibians only have one homologous structure means that it diverged at one point to form its own lineage, right? Right here. Okay. And then since these two share both the homologous structures, they both come down the same path, the same lineage, until we get to a point where we diverge both of them as well in our last part of the drawing, which is indicating that they separated as well. So it doesn't matter which one goes which place because since there's no other information given, you can replace marsupials here or placental mammals here. That's fine. But what's important is that you show that they did diverge from each other because they are different classes of animals, right? Okay, so it's step by step, right? First, identify the outgroup by determining which one has the least or no homologous structures between each other. That means that it would have diverged super early on at the very root, right? So we have our bony fish and our amphibians, marsupials, and placental mammals. Since they share homologous structures, they are most likely going to be under the same lineage, right? And next is putting more detail into it by determining which one is basically the odd one out, which one has less number of homologous structures. They indicate that they would have diverged earlier on in the lineage to become their own lineage. That's where we have amphibians. And then eventually we get to our last two, and you need to basically indicate that they diverge from each other as well in our last drawing. All right, continuing on from where we left off yesterday, we were talking about how to construct a phylogenetic tree. And the key thing to remember about this is that you always try to identify which species does not have any homologous structures with any other species. That indicates that it's going to be the outgroup, right? Basically, the species that diverge very early on from the root. So this is your root. The species that diverge very early on would go very early on would be your outgroup, and that would be the one with no homologous structures. That would be your bony fish. Right? And then we look at the species that have some homologous structures, but not all the same. That would be a species that diverged at one point from the main lineage. Okay, you have to realize that the outgroup is one lineage because it's separated early on, and then this is called the main lineage. This is where you're going to be working it from, right? So depending on the type of phylogenetic tree that you're constructing, whether you're constructing the conventional family tree shape, or whether you're using this shape, you need to know how to actually accurately draw it out. Okay, so we know that amphibians have one homologous structure in common with marsupials and placental mammals, but not the other one. So they would have diverged at some point to become their own lineage, right? And then finally, we have marsupials and placental mammals, and we know that you can't have one leaf having two species, so they would have diverged as well. But you have to indicate that their divergence happened very recently because they still share a lot of homologous structures. Okay, that is the key way to do it here. But one thing to note is this is for constructing using homologous structures. But questions might ask you, why are homologous structures no longer the standard to construct phylogenetic trees? That's because we are using molecular homology, right? Basically, we are studying the genes, studying the amino acid sequence, the amino acids that make up protein for a particular protein that's conserved, and or we are looking at their entire nucleotide sequence and then looking at the number of differences that they have, and then based on that we can also construct a phylogenetic tree as well. We're not going to practice that because I don't really have a question with that, but I'll try to get one of those questions during the revision class. But basically, using homologous structures is something we used to do very commonly, but when we started to develop our gene reading technology, we realized that by studying the genes, we can more accurately see the changes that have happened over time, and we can accurately describe it through a phylogenetic tree. So if they ask you what is the most reliable way to determine the evolutionary relationship between different species, your best answer would be molecular homology using either amino acid sequencing or nucleotide sequencing, and then comparing that with each other to see how many differences there are. And as we remember, if there are a lot of differences, that means they would have diverged earlier. If there are very little differences, they would have diverged very recently, right? And why is it that amino acid sequencing can be unreliable or not the best technique to use with closely related species? Can I get Yasmin? Why is amino acid sequencing not the best technique when we're dealing with closely related species such as chimpanzees and humans? So think about protein like hemoglobin, right? When we think about hemoglobin, between chimpanzees and humans, yeah, they're very conserved. Okay, you have to use certain words to best describe it. If you use the word same, it's not really accurately describing that they are conserved between each other. Okay, so make sure in science when we're trying to say that they're the same, you can use the word conserved. That means that between changing from one species to the other species, that specific sequence was conserved in the secondary sequence, the secondary species, right? So yeah, so at that point, we would actually use nucleotide sequencing to best understand the evolutionary relationship between the closely related species. Okay, so moving forward, getting into what we left off from yesterday. So yesterday we were looking at constructing phylogenetic trees, and we understood that they usually take a very easy to describe manner. But one thing to note is that there can be uncertainties in a phylogenetic tree because studying fossils and studying genes, studying molecular homology is not 100% technique to be able to determine the relationships that different species have had or the evolutionary relationships that they had. Because for one thing, fossil records can be inaccurate for multiple reasons. For one thing, uh, sometimes the fossils are not well preserved because the conditions for fossils to be well preserved are very specific. One, it needs to basically be buried under sedimentary rock in the right way, and it needs to be compacted in the right way, and then it needs to basically be preserved in the right conditions. So temperature, oxygen availability, all sunlight availability, all these play a factor in how well preservation of a fossil can occur. And if even one of those conditions is not perfect, then the fossil might not be as well preserved, which can be an issue, right? If we basically think about this in this way, right? If we never found the fossil for S, we would have never known that there was a divergence that happened that could be leading to another lineage altogether, right? Or possibly another example. Let's see if we can find another. Yeah. Let's say we are trying to find the fossils for the common ancestor between Ethiopian wolf, coyote, and dog. But when we are trying to find fossils, we could not find any fossils that accurately describe the common ancestor we're looking for. Thus, it creates a gap between what we know are related to each other and how they link back to the main tree, right? So we can't always fill in the gaps. In those cases, what happens is that there can be gaps in the phylogenetic tree that does not connect to the main branch to show how there's an evolutionary relationship. Instead, we know that they are closely related to each other through molecular homology, but we can't accurately describe at which point they would have diverged or from what main lineage they would have diverged from, right? Another thing, sometimes molecular homology is also not going to be accurate due to the fact that extinct species do not preserve their DNA as well, right? If you've watched Jurassic Park before, any of them, they always describe how we had to fill in the gaps of the genome with other DNA. That's because DNA degrades over time. It's not going to be preserved forever. In fact, if it's not preserved properly, it won't even last the first few months or years, right? So sometimes molecular homology can be very hard to use because there can also be barriers to be able to identify them. So all these uncertainties can be expressed in two main ways. One is that there's going to be a lack of a node between a species, indicating uncertainty of the divergence point. Specifically, we're looking at species Y. So we don't know at which point it would have diverged from the lineage. So what we end up doing is that we show that there is a branch leading to species Y, but we don't connect it to have a node with the main lineage, right? That is because we basically can't find the evidence of a transitional fossil between the two fossils, so they are left out. Basically, we can't find a transitional fossil that shows the transition from the main lineage into a divergent species, right? That is why we leave it out as a nodeless branch. Next is the when nodes usually split into two lineages or can even be, can split into three or more lineages rather than just two. So what does that mean is that essentially most of these lineages you see they split into two, right? One common ancestor diverged to become into two different species, right? But what can happen is that sometimes there can be even a divergence where three different species are emerging from one common ancestor, right? This is an example of adaptive radiation, which is talking about okay, which is talking about when there is basically a rapid divergence of different species happening from one common ancestor. And this can occur when there is actually insufficient data of sorry, insufficient data in relation to the two speciation events that occurred. Meaning that maybe that you would have diverged from B, but we don't have enough data to support that through our research. So what happens is that essentially it joins together with the separation from the main lineage, thus becoming a adaptive radiation event, right? So this is essentially when we are unclear which species it diverged from the others first, right? It could have been B that diverged from U, or could be U diverged from V, or T could have diverged from any of them as well. So it's unclear. So what we can do is just join them together at the same node, just to indicate that they are very closely related to each other. But other than that, we can't tell from which species it would have actually diverged from, right? And lastly is when there are one more weird case with a phylogenetic tree is the interbreeding event. So remember how I talked to you guys about Homo sapiens and Homo neanderthals and how they share DNA? So it's set in the sense that Homo sapiens until today who are not from Africa are having 2% of Homo neanderthal DNA within their own genomes. And this is theorized to have happened due to an interbreeding event between two different species that diverged super early on, right? So we had a common ancestor, probably the genus Homo at the early point, and then Homo neanderthals diverged from there, and then only we diverged from a different lineage to become into Homo sapiens. But what happened is that, so basically, this is the what's the word? The migration event that happened, right? Where a certain population of Homo sapiens moved into Europe and a certain population stayed in Africa. And what happened here is that this population that moved to Europe were overlapping with Homo neanderthals in the same habitat, in the same area. And there was an interbreeding event that happened. And at that point, we shared some of that genetic material, but you don't really need to indicate anything other than the fact that there is a interaction between them through an interbreeding event, but that can be represented by a line between the branches. Okay, you don't really deal with a lot of these exchanging genetic information between different species a lot. So this is a rare case, but you need to be able to identify what they mean if they just show you that in a question, right? If they draw a line directly between two different lineages, that means that it's talking about a interbreeding event where genetic material is passed between